Welcome, deep divers. Today we're plunging into something, well, incredibly fundamental, how our bodies manage fuel. It's like an intricate dance inside you.
It really is.
Our mission, really, is to unpack the mechanisms that keep your energy stable, even when, let's be honest, food intake can be all over the place.
Yeah, wildly variable sometimes.
And we're focusing on the two big players, the master regulators, insulin and glucagon.
Absolutely central.
And here's why it matters to you. Understanding these hormones is just crucial for grasping conditions like diabetes, hypoglycemia, things that affect millions. It's a window into your own biochemistry.
Exactly. Every single cell, every cell, needs that constant flow of ATP, the energy currency.
Right.
And the body achieves this through what we call metabolic homeostasis. It's this finely tuned balance intake, use, storage, mobilization. It's all about keeping that ATP supply steady, drawing on core concepts like those in Marks Basic Medical Biochemistry.
Okay, let's unpack that term a bit more. Metabolic homeostasis.
Yeah.
It's this precise balancing act you mentioned. Fuel coming in, getting burned, excess being stored, and then pulling from storage when needed, like a well-run power grid.
That's a great analogy.
So what controls this grid? You mentioned regulators.
Right. There are three main things keeping it balanced. First, the actual concentration of nutrients, like glucose or fatty acids, in your blood.
Okay.
Second, hormones like our stars today, insulin and glucagon.
The chemical messengers.
Precisely. And third, your central nervous system ships in too, sending signals. For example, the level of fatty acids circulating can tell your muscles, hey, burn fat now, save the glucose.
Interesting.
And within all this, glucose holds a really special place.
Why glucose specifically?
Well, some tissues are just completely reliant on it or heavily favor it. Think your brain, red blood cells, parts of your kidney, even your muscles when you're exercising hard. They need glycolysis breaking down glucose for energy.
So the demand is high.
Oh, yeah. Your brain alone chews through about 150 grams of glucose a day. Add another 40 grams for those other tissues. We're talking nearly 190 grams daily. If that supply falters, things go wrong fast.
Go wrong sounds pretty serious. Is there any wiggle room? Or is it always a crisis if the balance tips? What are the dangers?
Good question. The body is resilient. It has buffers. But push it too far, and yeah, it becomes a crisis. Let's look at both extremes. First, hyperglycemia, too much glucose in the blood.
High blood sugar.
Right. Chronically high levels can cause symptoms like polyuria, that's peeing a lot, and polydipsia, being really thirsty. Your body's trying desperately to flush out the extra sugar.
Okay.
But long-term, sustain high glucose. That leads to hyperosmolar effects, basically. Water gets pulled out of cells, which can cause serious neurological problems, even coma.
Wow.
And your kidneys have a limit, a threshold, for reabsorbing glucose. Go above that, and sugar spills into your urine. That's a classic sign.
What about the long term?
That's where non-enzymatic glycosylation comes in. Excess glucose literally starts sticking to proteins, changing how they work over time. The famous one is HbA1c.
The diabetes marker.
Exactly. When we see someone like Deborah S with an HbA1c of 9.5%, it tells us her glucose control has been poor for, the last few months.
And that leads to?
Serious complications. Microvascular, affecting small vessels, think eye damage, retinopathy, kidney damage, nephropathy, nerve damage, neuropathy, and macrovascular, bigger vessel problems like coronary artery disease, strokes, peripheral artery disease, atherosclerosis. It accelerates all that.
So high sugar is bad news long term. What about the flip side? Too low.
That's hypoglycemia. When blood glucose drops below roughly 60 milli-GDL, the brain really suffers. Remember, it loves glucose.
Right, the 150 grams a day.
Exactly. So you get neuroglycopenic symptoms, literally lack of sugar to the brain, like Connie C, feeling fatigued, confused, blurred vision. That's the brain running low on fuel.
Sounds scary.
It can be. If it's severe or prolonged, it can lead to loss of consciousness, seizures. It needs to be corrected quickly.
Okay, this is where it gets really interesting. Let's bring in insulin, the anabolic hormone, right?
Yep.
All about building and storing.
That's the one. Insulin is the primary signal for your body to store fuel and grow. It promotes glucose storage as glycogen in the liver and muscle.
That's compiling energy.
Yep. It drives the conversion of glucose to fat triacylglycerols in the liver, which then gets stored in your fat tissue.
Long-term storage.
Right. And it boosts protein building too. Increases amino acid uptake in muscle, helps the liver make proteins like albumin, and crucially, it actively stops the body from breaking down stored fuel. It's the, hey, we've eaten, let's store this bounty hormone.
How's it something so important made and released it must be tightly controlled?
Oh, it's a beautiful process. Insulin's a polypeptide hormone made in the beta cells within the pancreas, the islets of Langerhans. It starts as pre-proinsulin, gets processed to proinsulin in the ER that folds up, forming these vital disulfide bonds like molecular staples, giving it the right shape.
Uh-huh.
Then, in storage vesicles, an enzyme snips off a piece called the C-peptide. What's left is active insulin, ready to go. It even gets stored with zinc ions packed tightly.
So the cell's loaded and ready. What actually triggers the release? The doorbell, so to speak.
The main doorbell is blood glucose itself, but the way it rings the bell is clever.
How so?
Glucose enters the beta cell through specific transporters, GLUT2. Inside, an enzyme, glucogenase, traps it by adding a phosphate group.
Right.
This kicks off metabolism inside the beta cell, generating more ATP energy. And this rise in the ATP to ATP ratio is the key signal.
Energy levels rising.
Exactly. That high ATP closes special potassium channels, ATP-dependent ones. Less potassium leaving depolarizes the cell membrane.
Changes the electrical charge.
Yep. And that opens voltage-gated calcium channels. Calcium rushes into the cell.
And calcium is the trigger.
Calcium is the final trigger. That surge of intracellular calcium tells those vesicles full of insulin to fuse with the cell membrane and release the insulin into the blood.
Wow, that's quite a chain reaction.
It is. And it's tuned perfectly. The threshold is around 80 milligdl glucose, and release increases only when needed.
Amazing precision. Are there other signals besides glucose? Can't be just sugar telling it what to do.
You're absolutely right. Glucose is king, but other things modulate it. The autonomic nervous system helps coordinate things. Certain amino acids can give a little nudge too, though less potent than glucose.
Okay.
And interestingly, hormones from your gut, like GIP and GLP-1, released when you eat, actually give the beta cells a heads up, priming them for insulin release even before glucose spikes.
A preparatory signal.
Exactly. And conversely, epinephrine, the stress hormone, actually hits the brakes on insulin release. In fight or flight, you need energy available, not stored.
Which brings us right into the clinical side. Problems here are central to major diseases.
Absolutely. Think of Dianne A. with type 1 diabetes. Her immune system attacked and destroyed her beta cells. She has virtually no insulin production, hence the need for injections.
Total deficiency.
Right. Then there's MODY type 2. It's a genetic thing, a mutation in that glucokinase enzyme we mentioned.
The one that traps glucose in the beta cell.
Yes. With a less active enzyme, you need higher glucose levels just to get the insulin release process started.
So a higher threshold.
Exactly. And there's neonatal diabetes, sometimes caused by mutations in that potassium channel gene. If the channel stays open when it shouldn't, the cell can't depolarize properly and insulin release is impaired.
Fascinating how one tiny channel protein is so critical.
Isn't it? And then there's Connie C's case, the insulinoma. That's a tumor of the beta cells just churning out insulin nonstop, regardless of blood sugar.
Leading to her low blood sugar, the hypoglycemia.
Precisely. And remember, the C-peptide. Measuring it alongside insulin and glucose is really useful for diagnosis. Since it's released with insulin but hangs around longer, it tells doctors how much insulin the patient's own body is making. Crucial if they might be on insulin shots already.
Okay, so insulin is the store signal. Let's switch gears.
Yeah.
What about the release it signal?
Glucagon, right. Glucose is gone, hormone. Perfect transition. Yes, glucagon is the major hormone telling your body to mobilize stored fuel. Its main targets are the liver and adipose tissue.
Not muscle.
Critically, no. Muscle cells don't have glucagon receptors. So in the liver, glucagon kicks off glycogenolysis, breaking down stored glycogen to release glucose.
Okay.
And it ramps up gluconeogenesis, making new glucose from things like amino acids, lactate, glycerol.
So tapping reserves and making new fuel.
Exactly. And in fat tissue, working alongside lower insulin levels, it triggers the release of fatty acids from storage, provides an alternative fuel.
How is this hormone made and controlled? It seems like it needs to be just as responsive as insulin, but in the opposite direction.
It does. Glucagon's also a polypeptide made in the alpha cells of the pancreas. It's processed from a precursor, pre-pro-glucagon. It acts fast and is broken down quickly. Its half-life is only about 3 to 5 minutes.
Short acting.
Very. Ensures tight control. The main signals for its release are a drop in blood glucose and/or a rise in insulin. Actually, insulin directly suppresses glucagon release from neighboring alpha cells.
Direct communication within the pancreas.
Yes. So glucagon is lowest after a carb-heavy meal and highest during fasting. But here's a neat twist, the amino acid paradox.
Paradox.
Many amino acids actually stimulate glucagon release. Think about a high-protein meal. Insulin goes up to help muscles take in amino acids, but that could cause low blood sugar.
Right.
The simultaneous glucagon release prevents that dip in blood sugar and makes sure the liver has amino acids available if it needs to make glucose. It's a safety mechanism.
Clever. So how does glucagon get its message across inside the target cell? Another complex relay.
It is, and it beautifully illustrates how these signals work. Glucagon binds to its specific receptor on the liver or fat cell membrane.
Step 1.
This activates G proteins inside, which then switch on an enzyme called adenylate cyclase.
Correct.
Adenylate cyclase churns out cyclic AMP, or CAMP. That's the key intracellular second messenger. It amplifies the signal massively.
Like an internal megaphone.
Great analogy. CKMP then activates another enzyme, protein kinase A, or PKA.
Master switch.
Kind of.
Mhm.
PKA is the enzyme that does the phosphorylating, adding those phosphate switches to other key enzymes. This activates some pathways like glycogen breakdown and inhibits others like glycogen building. It orchestrates the whole response.
And you said it's short-lived.
Yes, CKMP is rapidly broken down by phosphodiesterase. This means the signal stops quickly when glucagon levels fall, allowing for really dynamic control.
So looking at glucagon's pathway, what are the big principles of hormone signaling it shows us?
Several key things. Specificity of the receptor means only target cells respond. Liver, yes. Muscle, no. Amplification one hormone molecule triggers a huge downstream effect via CKMP and PKA.
Right.
Integration PKA coordinates multiple pathways simultaneously to achieve the goal, like mobilizing glucose.
Makes sense.
Then augmentation antagonism signals can add up, like glucagon and epinephrine both boosting CAMP, or they can oppose each other. And finally, rapid termination breaking down CAMP quickly allows the system to respond to changes fast.
It's incredibly elegant. Are there everyday things that mess with this CAMP pathway?
Oh, definitely. Think caffeine.
My morning coffee.
Exactly. Caffeine is a methylxanthine, and it inhibits that phosphodiesterase enzyme.
The one that breaks down CAMP.
Right. So caffeine makes the CAMP signal last longer, kind of mimicking glucagon's effects, promoting glycogen breakdown and fat release. A little biochemical boost.
So my coffee is giving me an energy kick, biochemically speaking. Beyond insulin and glucagon, are there other players in this constant balancing act, especially hormones that work against insulin?
Yes, we have the insulin counter-regulatory hormones. The big ones are epinephrine and cortisol.
Fight or flight and stress hormones.
Basically, yes. Epinephrine is a catecholamine, acting super fast. It preps you for stress, mobilizes fuel quickly, ramps up heart rate, blood flow.
How does it signal?
It binds to adrenergic receptors. Beta receptors, like in the liver and muscle, use that same CAM-MP system as glucagon to trigger glycogen breakdown. Alpha receptors use a different pathway, but also boost glucose release from the liver. It's all about immediate energy availability.
And cortisol.
Cortisol is different. It's a steroid hormone, a glucocorticoid. It doesn't bind to surface receptors. It goes inside the cell, binds to a receptor there, and the whole complex goes into the nucleus to change which genes are being turned on or off.
So slower, but maybe longer lasting effects.
Exactly. It takes hours, but the effects persist. Cortisol often works with glucagon, for instance, by boosting the production of enzymes needed for making new glucose, gluconeogenesis. Chronic stress, leading to chronically high cortisol, can actually contribute to glucose intolerance over time.
It really sounds like diabetes isn't just an insulin story then. You mentioned a bihormonal aspect.
That's a crucial point. In both type 1 and type 2 diabetes, even though blood sugar is high, glucagon levels are often inappropriately elevated too.
Why?
In type 1, it's because there's no insulin to suppress the alpha cells making glucagon. In type 2, the alpha cells themselves can become resistant to insulin's suppressive effect. So this extra glucagon just makes the high blood sugar situation even worse. It's a breakdown in that crucial insulin-glucagon balance.
Okay, let's bring this all home. Let's revisit our patients to really see these mechanisms in action.
Great idea. Let's start with Deborah S, type 2 diabetes. Her core issue is insulin resistance. Her cells, especially muscle and fat, aren't responding properly to the insulin her body is making, often linked to obesity, especially belly fat.
So the signal isn't getting through properly.
Right, often a problem downstream from the receptor. That's why drugs like sulfonylureas, like glipizide she might take, can help. They work on the beta cell's potassium channel, forcing it closed to kind of squeeze out more insulin, helping to overcome that resistance somewhat.
Okay. And Dianne A, type 1.
Pure insulin deficiency? beta cells destroyed. And remember, C-peptide. Since injected insulin doesn't have it, measuring Dianne's C-peptide tells us if her body is making any insulin at all. Very low or zero C-peptide points strongly to type 1.
A clear diagnostic tool.
Definitely.
All right.
And finally, Connie C with the insulinoma. Her symptoms, the dangerous lows, the confusion, the shaking, sweating, racing heart that was all driven by her tumor pumping out massive amounts of insulin uncontrollably.
And the fix?
Surgical removal of the tumor. It was full of those rogue beta cells. Removing it stopped the excess insulin secretion and her symptoms resolved. It's a direct link between uncontrolled hormone release and severe metabolic disruption.
These cases really illustrate the principles. So, wrapping this up, what are the key takeaways for you, our listener, from this deep dive?
I think there are a few big ones. First, keeping blood glucose stable isn't just important, it's absolutely vital for survival, especially for your brain.
Non-negotiable.
Right. Second, insulin is the master of storage, glucagon the master of mobilization. They're in constant, delicate balance.
The yin and yang of metabolism.
Kind of. Third, those signaling pathways. The CAMP cascades, the phosphorylation, that's the language hormones use to communicate rapidly and effectively throughout your body. Keeping glucose in that tight range, ideally 80-100 milli-GDL. And finally, when this system breaks down not enough hormone, cells not listening, or rogue production, you get serious diseases like diabetes and hypoglycemia. Understanding the basics really illuminates the clinical picture.
And knowing these core biochemical concepts really is foundational, isn't it? It helps make sense of health advice, disease processes, even just how your own body feels day to day.
Absolutely. It empowers you.
So here's a final thought. Next time you eat a meal or feel that rush of adrenaline during stress or exercise, take a moment. Consider that incredibly complex, usually invisible symphony of insulin, glucagon, epinephrine, and others working tirelessly inside you. What subtle signals might your body be sending about its fuel state right now? Thank you for joining us on this deep dive into fuel metabolism.
Thank you. It was a pleasure.